Hello everyone, my name is Darshini Satyamurthy from DBT3A. My metric number is 23 DBT20F1025. Today I am going to do a lab simulation based on bacteria identification. Before I start the lab simulation, I want to explain about bacteria identification. Bacteria are identified routinely by morphological and biochemical tests, supplemented as needed by specialized tests such as serotyping and antibiotic inhibition pattern. Newer molecular technique permit species to be identified by their genetic sequence, sometimes directly from the clinical specimen. Now enter the lab. Welcome to the Virtual Bacterial Identification Lab. The purpose of the lab is to famili- familiarize you with the science and technique used to identify different type of bacteria based on their DNA sequence. Not long ago, DNA sequence was a time-consuming, tedious process. With readily available commercial equipment and kits, it is now routine. The techniques used in this lab are applicable in a wide variety of settings, including scientific research and forensic lab. The basic step is below. The first one is prepare a sample from a patient and isolate whole bacterial DNA. The second one is make many copies of the desired piece of DNA. The third one is sequence the DNA. The fourth one is analyze the sequence and identify the bacteria. The piece of DNA used for identifying bacteria is the region of code for all small subunit of the ribosomal RNA. We will refer to this piece of 16 as rDNA. Different bacteria species have unique 16 as rDNA sequence. The identification relies on matching the sequence from your sample against a database of all known 16 as rDNA sequence. The learning of objective is what kind of patient samples are used for the purpose of identifying possible pathogens. The second one is what does PCR do, how does it work, and why is it useful. The third one is how do you separate the desired DNA from all others. The fourth one is how does an automatic DNA sequencer work. And the last one is why is it possible to use a DNA sequence to identify bacteria. Throughout each exercise, this window will display information, explain what you are doing. All the interaction, however, will be done inside the graphic window to the left. The small white box below the graphic will give you specific instruction on what objects to click on. Now, our record show that you have visited the virtual bacteria ID lab before. If this first time using the lab, click the start over button below. The part 1 is sample preparation. The key to a successful identification is to start with a good sample. Many pathogenic bacteria do not grow well on solid culture medium, making identification by traditional means difficult. Even poorly growing bacteria culture, however, can be identified with the methods used in this lab. In this lab, I will act the pathologist or perhaps a pathology lab technician at well-equipped research hospital. Our task is to identify a bacteria sample received from a clinician. Welcome to the Virtual Bacteria ID Lab. To begin, please put on a pair of rubber gloves by clicking the glove box in the open drawer. Now I will obtain a sample from the culture dish. Click the wire loop to continue. Click one of the bacterial colonies in this dish. Click the microcentrifuge tube to transfer the bacteria colony from the wire loop into the microcentrifuge tube. Planning bacteria colony into the microcentrifuge. The second part is PCR amplification. The step 1 is add master mix. To prepare the polymerase chain reaction PCR, we will add the PCR master mix solution to our sample DNA. The PCR master mix solution in the red cap bottle contains the following: water, a buffer to keep the mixture at the correct pH for the PCR reaction, large quantity of the 4 nucleotide adenine, cytosine, guanine and thymine, large quantity of oligonucleotide DNA primer that bind the 16S rDNA region to initiate the replication process. The second step is run PCR. Once the reaction tube are loaded onto thermocycler, the PCR machine, the automatic process of DNA replication starts. Refer to the animation. The machine used in this lab has read out the describe what is happening. I will now add the PCR master mix solution to all tubes. Click the red cap master mix bottle to continue. The third part is purify PCR product. If you if you are confident that the PCR work, you can proceed to purifying the PCR product. Running a gel is actually one method of purification. Once the PCR product is in the gel, you can cut out the band corresponding to the PCR product and isolate the DNA from the gel. Nowadays, you can buy compact microfilter to filter the DNA from the PCR tube without running a gel. The fourth part is prepare for sequencing. The DNA sample has been purified. Your PCR tube should now contain almost nothing but copies of the R16S rDNA. Now we can prepare the sample for automatic sequencing. DNA sequencing technology is another area of molecular biology that has seen an impressive amount of refinement. The, the predominant method illustrated here is called PCR cycle sequencing. Firstly, I will dilute the PCR product to increase the volume. Click the blue cap distilled water bottle to continue. The strip tube has been loaded. Click the PCR mission to continue. Amplification. Click above to pause or replay the animation. Click done when finished. Each green and blue tube contain a sequence brew consists of buffer, primer, a different one in each tube, DNA polymerase nucleotide and fluorescence tag terminate in suitable proportion. The PCR product from the previous step is added to each tube and another PCR is run. This time the aim is not produce identical copies of DNA but many copies of variable length. The animation illustrates what is happening in one tube contain the primer 65 1C 1R. Each DNA strand bind the primer at one end and will have the fluorescence tag terminator at the other end. Now click the Done button when done the PCR amplification. The sequence preparation is complete. Click the orange tray. Continue to Part 5 DNA sequence. Moreover, now Part 5 DNA sequence. From the last step, you have 12 tubes that contain the final PCR product, a mix of DNA pieces of variable length. All DNA pieces in each tube start with the same primer but end with a different nucleotide tagged with a fluorescent marker, different color for each nucleotide A, T, G, C. What remains to be done is to separate the individual DNA pieces and identify the end of nucleotide. This is done by using an automatic sequencer that performs gel electrophoresis on the DNA in each tube. Gel electrophoresis is a method to separate molecules based on difference in size. The sequencer used in this lab has a thin capillary tube attached at one end and to syringe mechanism that contain a buffer solution. The tube is filled with the buffer solution and other and insert into one of the tube containing the DNA pieces. Then an electric current is applied so that the end of the tube is contact with the DNA has a negative charge and the syringe end a positive charge. Since the DNA molecules are negatively charged, they start to move through the tube toward the positive charge syringe end, with the smaller pieces moving faster than larger one. Near the syringe end, the capillary tube passes through a laser beam that excites the fluorescent markers and optical detector detect the color of the fluorescence. We can assume that the complete set of DNA pieces, all differing in size by exactly one nucleotide, were generated in the previous step. The smallest piece of DNA has fluorescent tag attached to it is the primer. This DNA fragment will travel faster than the other one. The sequencer automatically flushes out the buffer from the tube, move the tray and run the electrophoresis again, repeating the program until all 12 tubes have been examined. Now sequencing, click above to pause or replay the animation, click done when finished. Now click the Done button. Now I have completed the DNA sequence section. Click the orange tray to continue to part 6. Part 6 Sequencing Analysis. Finally, here is Part 6 DNA Sequence Analysis. The sample A is fluid from lymph node. After the sequence information has been gathered from all the reaction tube, the computer built up the actual sequence matching together different pieces. I now have the 16S rDNA sequence from this bacteria species, which can be compared with all other known 16S rDNA sequence from identification. Congratulations, I have correctly identified the bacteria. This sample was obtained from tissue and fluid aspirated from lymph node of a patient complaining of a fever, fatigue and swollen lymph gland. The sample was chemically treated and placed on agar that support the growth of bacteria. Petri dish contained clearly visible colonies of bacteria were sent to the laboratory for identification of the specific type of bacteria by PCR amplification and sequence analysis. The Bartonella hanselii is various species of Bartonella that are pathogenic to humans are transmitted via a vector or directly from an animal reservoir. For example, B. bacilliformis via sand flies causes Orea fever. B. quanthana via body lie causes trans fever, and B. hanselii via cat causes cat scratch disease (CSD). CSD typically manifests as swelling of the lymph gland, possibly with skin lesion at the site of inoculation and possibly accompanied by fever, fatigue and other symptoms. These are may be particularly susceptible and can develop a different disease. That's all from me. Thank you.